Okay, good afternoon, everyone. I think we will go ahead and get started with our presentation today. Welcome to the MyHorse University and University of Minnesota's live webcast titled Advances in Equine Genetics. This presentation is the fifth of 7 webcasts offered in a series to bring you the most up-to-date information on equine genetics. This series is funded by the United States Department of Agriculture's National Research Initiative. Future webcasts will focus on EMS and ponies and other genetics. Our presenter today is Dr. Molly McCue, an assistant professor in veterinary population medicine at the University of Minnesota College of Veterinary Medicine. She graduated from Kansas State University with a bachelor's degree in veterinary medicine and animal science in 1998, a DPM degree in 2000, and a master's degree in equine internal medicine in 2004. In 2007, she earned her doctorate in comparative and molecular biosciences from the University of Minnesota. Please note that you are able to ask questions during the presentation via the text chat to the left of your screen. And our presentation today will be recorded and uploaded to our website if you'd like to review it at a later time. And at this time, I will turn our presentation over.
Thanks, Amanda. And thanks, everybody, for joining us today. So the title of the presentation, Advances in Equine Genetics, is a little bit generic. But what I really want to focus on are really the direction that I think that equine genetics is moving and what people need to know about interpreting genetic tests. So if you've tuned in for a previous genetic webcast, we've talked a lot about simple gene traits, and I'm going to review a little bit of that today, but really I want to kind of move forward from there. So just an overview of where we'll be today. Again, a brief review of genetics and the terminology for people who haven't been listening to prior webinars, and just even for those of you who have, just to make sure that we're all on the same page and using the same terms. And then really after that, switching over to what I think the future of equine genetics is. We've hinted on this in previous webinars with really increasing emphasis on complex traits. And then also the fact that we used to think traits that were simple, particularly in disease genetics, we're realizing aren't as simple as they used to be. So I want to talk a little bit about that and walk through a couple of examples of what complex traits are and some of these simple Mendelian traits that are turning out to not be so simple. And then the last thing I want to touch on is just a little bit about genetic variation in the horse and genetic diversity. So with the new genome tools, we have a chance to really explore these things in a way that we haven't in the past. And it's something that the equine genome community, as far as the research side, is focusing on more and more. So I thought I'd touch just a little bit about what that was and where that was going. So again, for review, in the equine genome, it was sequenced in 2007. The first draft sequence was out in February, and from that we know that horses have about 22,000 equine genes. This is pretty similar to the count in people and other mammals. We knew previous to sequence that it was arranged on 33 chromosomes, but now we know the exact location of those genes and their exact sequences. And those 33 chromosomes are, 31 of them are autosomes, and then there's the X and Y, the 2 sex chromosomes. Remember that while there's 33 distinct equine chromosomes, each one of them is paired. So an individual actually has 2 copies of each of the 31 autosomes and then has either 2 X's or an X and Y depending on gender. So each copy of a gene is called an allele. An allele is a term that we'll talk about a lot through this webinar, and important to remember because there are more than one type of allele that can be present on a gene. So I have an example here of an individual that has a paired set of chromosomes. For example, let's say this is equine chromosome 1. One copy will come from the sire and one copy will come from the dam. And so I have listed the genes here, A, B, and C, represented on this chromosome, and you can see that the A allele from the sire is slightly different than the A allele from the dam, noted here by a capital A and a lowercase a. And so understanding that while this is the same gene, the sequence of this gene can be slightly different in the way that it's inherited from the sire and the dam, and those changes in gene sequences are important in understanding genetics and genetic variation. So the different alleles of a gene code for different variations in proteins, and so for the purpose of this webinar, I'm really going to focus on genes as protein coding genes, so genes that result in a change in protein, which are really the #1 building block in the body. There are genes that do other things, but the majority of the ones that we're concerned about, and for simplicity's sake, are protein coding genes. So all horses have a combination of alleles at a single locus, at a single gene, and that's called the horse's genotype. And genotype is pretty simple. With technology that we have today, we can actually look at gene sequences in individual horses at a locus, and we can call specifically what its genotype is. An individual's phenotype is what you see as a result of the genotype. So I've put in a simple example here. We've got a gray horse, and if we were, for example, to genotype this horse at the gray locus, and his genotype was NG, meaning one normal allele and one gray allele. His genotype is NG, but his phenotype is actually gray. And there's, we'll talk about this again in a moment, but there's a couple different combinations that can make you genetically gray. So there's not always a complete one-to-one correlation between your genotype and phenotype. And that's something that I really want to try to focus on in this webinar and try to explain and drive home that while we can always, almost always based on sequence, tell you what an individual's genotype is, depending on the genes, it may or may not be simple to predict what the individual's phenotype is going to be. And that's really the overarching difference between simple genetic traits and complex genetic traits, and a lot of the things that we're going to talk about from here moving forward. So the genotype to phenotype correlation that we've talked about in previous webinars, and the one that's most simple, are the Mendelian traits, or simple genetic traits. And so for the most part, simple traits mean that the relationship between genotype and phenotype is either dominant or recessive. So the last webinar we spent a lot of time talking about this, so just briefly review here. A recessive trait means that you have both copies of the gene must carry the mutation or a specific allele to cause a phenotype. So, for example, if you had an allele, capital A, that resulted in a trait of interest, say coat color, if it's recessive, you would have to have 2 copies of the A allele to have the trait. If you had 1 copy and then had B as your other allele at that gene, or 2 copies of the B, you wouldn't express the trait. A dominant trait works a little bit differently in that only 1 copy of the gene must have the allele to cause the phenotype. So in this example, the AA and AB genotypes would express the trait or the phenotype of interest, while BB would not. So simplest examples in horses come from coat color genetics, are the ones that we've known for a very long time. And I will say, if any of you are very familiar with coat color genetics, I'm simplifying a little bit, but let's say, for example, we're going to look at the chestnut locus. So the wild type color of a horse, the base color, is brown, which can be modified by other loci, but we're going to ignore that for the moment. And that is designated typically in equine genetics as the capital E for the extension locus. So if you're wild type capital E, or capital E lowercase e, you're going to be brown. But if you have the chestnut mutation, which is designated by the lowercase e, if you've got 2 copies in recessive manner, you're going to be chestnut or sorrel, depending on the breed's use of that color designation. Similarly, we talked a little bit about gray a moment ago. If you don't have any copies of the gray mutation, for example, we're looking within the Percheron horse here, they're typically either black or gray. No copies of the gray mutation, this horse happens to be genetically black, but gray is dominant over that base color. And so if you have 1 or 2 copies of the gray allele, you'll have the gray phenotype. And so just by looking at this particular gray horse, we can't predict his genotype. We know it's one of these 2 alternatives. But by looking at his genotype, because it's a simple trait, we can clearly predict what his phenotype would be, even without seeing the horse. So there's several known simple traits in horses, and again, we've reviewed these in previous webinars, so I'm not going to go into much detail, but the coat colors are probably the best known, what I call simple traits, in that when you take each one of them in and of itself, it's pretty easy to predict phenotype. That being said, I don't want to confuse anyone or certainly misspeak in that you have to take kind of the summation of all coat color loci together to know what the true coat color locus is. For disease mutations in horses, the simple mutations, the list is growing all of the time. HYPP, or hyperkalemic periodic paralysis, was the first one that was identified in the early 90s, followed by severe combined immunodeficiency. Overo-lethal white syndrome, the overo coat color mutation also can cause lethal white, so I don't have it listed in both places, but they're interrelated, and we'll talk a little bit about that in a moment. Glycogen branching enzyme deficiency, 2 forms of JEB, HERDA, polysaccharide storage myopathy, which we'll focus on a little bit more during this webinar, is a simple trait that's not so simple. And then some of the recent ones: lavender foal syndrome, dwarfism, congenital stationary night blindness, which is associated with the leopard complex coat color mutation in the Appaloosa, and Faulponi syndrome. And there's a few others that aren't published yet, but are recognized and will be out in the scientific literature, I would guess, within the next 6 months. So that's where we've been in equine genetics, and that's where we have focused previously in webinars, is really talking about the known mutations, what they look like, what the phenotypes are, particularly in our webinars with the emphasis on disease. But where we're heading is in a different direction, and again, this has been alluded to in the past, but with the sequencing of the equine genome research community was able to develop a lot of new tools that really are allowing us to rapidly identify new genetic traits in the horse and new genetic variants. So with this additional research, I think we're realizing 2 things. The first is that what we used to think were simple traits, particularly in disease genetics, are oftentimes more complicated than that, so interpretation of a genetic test takes several factors into account. And then 2, really moving forward, is the focus on complex genetic traits. And so in the equine research community, we're moving away from trying to discover single mutations that cause disease, but to really understanding these complex genetic diseases that may have many mutations underlying them. So what does that mean for the genotype to phenotype? Well, thus far, genotype to phenotype has been relatively simple, but it's not always that straightforward. So what can complicate this interpretation? One would be modification of the expression of a simple genetic trait by the environment. So this is kind of related to that nature versus nurture idea that people have probably heard about for a very long time, and that you can have a genetic trait, but you may not show the genotype unless you're in the right environment. We'll talk about a couple of examples of that. The other could be modification by other mutations or modification by other genes, and this is really probably the full story for coat color genetics, that they all can modify each other depending on the loci. Something else that we talk a lot about, which I think people struggle to understand, and even when speaking to veterinarians and talking about disease genetics and interpretation of tests, is this idea of incomplete penetrance, which means that you can have the genotype that's associated particularly with disease, but sometimes color or other characteristics, but not necessarily show the phenotype, and I'll come back to that in some examples. And then the final thing is pleiotropic effect, and I alluded to this a little bit earlier, but for example, OWLS, or overo-lethal white syndrome, if you have 1 copy of the overo gene, and you would express the overo coat color in a paint horse, but if you have 2 copies of this gene, you're actually born a lethal white foal and have some issues, and most notably lack of normal function of the GI tract, which is fatal, usually within a couple days from birth. The other example of a pleiotropic effect is congenital stationary night blindness. So the leopard complex, or the mutation that causes leopard spotting or the blanket spotting in Appaloosas, is also associated with abnormalities in the retina. So if you have 1 copy of that gene, you will have 1 particular Appaloosa coat color pattern. If you have 2 copies, you will be a different Appaloosa coat color pattern, but along with having 2 copies of the gene, you also have night blindness, meaning that you see fine during the day, but because of retinal abnormalities, these horses are basically blind at night. So we select for the coat color in both of these diseases, but when we inadvertently get 2 copies of the gene, we have what we consider a disease trait. So as an example of some of these things, I want to talk a little bit about polysaccharide storage myopathy. This is a disease that we've talked about in previous webinars. It's also a disease that we spend a lot of time researching in our lab. And in my mind, it's a good example of some of this idea that what at first seemed to be a simple genetic trait actually is a little bit more complex. So type 1 PSSM is caused by a dominant mutation in a gene that we call glycogen synthase 1. This mutation occurs in 25 different horse breeds. What happens in these horses, if they have 1 copy or 2 copies of this mutation, they actually start to accumulate excess glycogen in their skeletal muscle, and it's because the glycogen synthase enzyme works too well. So we think, based on muscle biopsy, that this looks like a dominant trait. There's a couple of interesting things that we notice about the trait right away. It's in 25 different breeds, which is unique to most genetic mutations in horses, and also that the phenotype, or the disease that we see in different breeds, is slightly different. It most commonly occurs in quarter horses and their related breeds, and also in draft horses. And interestingly, in quarter horses, the most common complaint is that these horses tie up. So they're asked to exercise, they get exertional rhabdomyolysis, or tying up, or muscle pain and stiffness, and they stop exercising. And they will do this repeatedly, not necessarily with every bout of exercise, but usually these horses have multiple episodes within a year or throughout their lifetime. It can be quite severe. So when, as a veterinarian, we're presented with these cases, the complaint is usually tying up or exercise intolerance. When we look at draft horses that have the same mutation, while they can tie up with this disease, we oftentimes see a little bit different manifestation. And so when we do a skeletal muscle biopsy, we see the same abnormal glycogen. We know these horses have the same mutation, so their muscle phenotype is not that different. But clinically, their phenotype is very different, and that draft horses usually kind of have muscle wasting. And if you can see in this picture, this horse has got some wasting over her top line. She's got some wasting here in her gluteal muscles, and maybe you can even see a little bit in the neck. And so they tend to have muscle wasting, and over time, these horses get weak, and they show muscle weakness. And so it's not necessarily that you ask them to exercise and they tie up acutely, but they definitely have some weakness in their skeletal muscle. So right off we see that the same genetic mutation in 2 different breeds, which we would term 2 different genetic backgrounds, can have a little bit different phenotype. So when we first identified this mutation, this was work out of our lab, we wanted to estimate what disease penetrance was based on the muscle biopsy. So if you look at the muscle biopsy and correlate that particular phenotypic measurement with the genotype, we see that homozygous horses, those with 2 copies of the mutation, 100% of the time have skeletal muscle biopsy abnormalities. And we see that heterozygous horses with 1 copy of the mutation, 95% of the time we can identify that they have a clear abnormality in their skeletal muscle. And so from that we conclude that this is a highly penetrant dominant disease. When we start looking at other phenotypes which are associated with PSSM, and specifically in this case clinical signs in quarter horses of tying up, we see that there are some things that affect the expression of phenotype. And 2 things that a long history of research has demonstrated is that with quarter horses that have the GYS1 mutation, if you feed them a low-starch, high-fat diet and you provide them with consistent daily exercise, you can actually dramatically improve their clinical signs, and in some individuals completely eliminate any clinical signs associated with this disease. So I have just a little bit of graph here to just explain this a little bit more. In this graph, we're looking at the y-axis serum CK, and CK is a muscle enzyme and an indicator of muscle damage. So the higher the CK value, the more muscle damage in a horse. And along the x-axis, we have days of exercise. And so this was a group of horses that were all heterozygous for the PSSM mutation that were put on a treadmill to actually do light exercise. And so as we exercise these horses at basically day 1, we checked their CK activity. They were a little bit higher than normal, about 25,000, whereas we expect 400 or below to be normal. And by day 2, we see a pretty significant spike in their CK. And then as we provide them exercise over time, even in as little as 8 days, we see that this starts to improve. So in this instance, we would say that the phenotype of PSSM as far as clinical signs is heavily influenced by the environment. If we go back and look at a muscle biopsy from these horses on day 0 or day 8, we'll still see the abnormal glycogen that's still present, but we're able to change their clinical phenotype by modifying their environment. The other thing that we saw in PSSM that was kind of interesting is that when we took a set of horses and we fed them high-fat diets and we provided them consistent daily exercise, not every single horse necessarily responded. And so in the previous slide, I had the number of 75% of horses responded. And then we also saw that the clinical signs that were demonstrated by these horses were really variable from one individual to another. Some horses, when asked to exercise, would just get a little muscle sore, a little stiff, a little bit of muscle cramping, whereas others would be so painful they wouldn't move. They would have so much muscle cramping that they would become recumbent or lay down and be unable to rise. And then in a few instances, we had horses that died acutely in the middle of a tying up episode. And so we started asking some questions about what else is it besides diet and exercise that might be modifying the clinical sign that we see in these horses. And one thing that we recognize going back and looking at clinical histories from about 500 horses, 500 to 600 quarter horses with PSSM, and looking at pedigrees, as we started to recognize that we had these large families of horses that were related that also were the ones that had very severe clinical signs. And so in this picture, we're looking at a pedigree from horses, and individuals with dark circles would be ones that are affected with PSSM. Individuals with an N within the circle would be ones that were normal. These individuals that have a little arrow would be ones that were ancestors or sires of dams of unknown several affected horses. And so we started asking the question, like I said, could there be something genetically that's also modifying the PSSM phenotype that we seem to see the most severely affected horses running in families? And so we did some genetic analyses in these families, and what we came upon is that within this family, not only did these horses have a GYS1 mutation causing polysaccharide storage myopathy, they also had a mutation in a gene called RYR1, which underlies malignant hyperthermia, which is another genetic disease that we see in quarter horses. Now, in and of itself, a mutation in RYR1 and the disease malignant hyperthermia is not something that we worry about very much clinically, because these horses typically only demonstrate clinical signs of malignant hyperthermia if they're anesthetized, and most horses are not anesthetized with inhaled anesthesia through their lifetime. Additionally, the mutation underlying malignant hyperthermia is very rare in the quarter horse population. About 1/2% of horses have it. But what we recognized within this family is that a large number of these horses, somewhere around 40% of them, actually had this malignant hyperthermia mutation. And when we looked at horses that had both the RYR1 and the malignant hyperthermia mutation together, we saw that they had more severe clinical signs. And so, again, I've just put a little bit of data in here on the graphs to demonstrate this. Now we're looking at CK again. These values are a little bit different in that we've log transformed them because they were so hugely variable in these horses. They wouldn't all fit on the graph. But what you can see is that the horses in red here are PSSM and MH horses. They have both mutations, and the horses in yellow have only PSSM. And as we exercise them over time, we see that really these horses with both mutations consistently have a higher CK. There's one day where they were pretty close together, but they're worse as far as a CK measurement as an indicator of muscle damage than these horses in yellow that have only PSSM. And furthermore, when we add the combination of a high-fat diet to these horses, they start out worse than the horses with only PSSM, but both of them get a little bit better with exercise over days. But while horses with one mutation, again, improve with a high-fat diet, they're still, even out here on the graph, we see that the horses with both mutations are worse. And we look at this in other ways. We know that these horses with both mutations can complete less exercise. So whereas PSSM only horses might be able to exercise for 20 or 30 minutes, these horses with both mutations can only exercise for 15. And so really a more severe clinical phenotype when both mutations are involved. So what kind of conclusions can we draw from this? Well, what this means practically to horse owners and practically for testing and genetic testing for veterinarians is if you have a horse with PSSM and you perform the genetic test, there's a couple of things that you have to consider. One is the genotype of the horse and what's the likelihood for them getting PSSM. And two, that if you modify the horse's environment by exercising them every day and putting them on a high starch diet, even if you have a horse that's heterozygous or sometimes homozygous from PSSM, you aren't necessarily going to see clinical signs of disease. The second thing to remember is that if you have a quarter horse that has PSSM, particularly those with very severe disease, it's probably important to look for the RYR1 mutation as well. And so the story, although PSSM is inherited dominantly based on muscle biopsy, when it comes to interpreting the clinical relevance of the test, is not quite so simple. So I want to come back again to this idea of this term of incomplete penetrance, because PSSM in some environmental conditions is an example of this. Penetrance basically is defined as the proportion of individuals that carry a particular allele or genotype that also express the associated phenotype. So in PSSM we talked about when the phenotype was measured as muscle biopsy, there was complete penetrance, but when the phenotype is measured as clinical disease, there's not necessarily complete penetrance, and so it's not a one-to-one correspondence every time. In medical genetics, particularly on the human side, penetrance is defined a little bit differently in that it's the proportion of disease-causing the penetrance of a disease-causing mutation is the proportion of individuals with a mutation that have clinical signs. So in my mind, it's a little bit simpler definition. So, for example, a penetrance for PSSM in particular environments where horses are exercised consistently and don't have a lot of starch could be as low as, say, 50% for heterozygous individuals. When we start talking about penetrance and we start talking about the fact that it's not complete, the terminology that's often used in human medical genetics and the terminology that we're starting to use in horse genetics is the idea of a risk allele. So a risk allele is an allele with incomplete penetrance and can be low to moderate. So we move from talking about a yes-no recessive dominant mode of inheritance to talking about the genetic risk associated with an allele. So what's an example of this? Another example from horse genetics. Well, disease risk and melanoma. So we know that gray coat color horses is caused by a dominant mutation in a gene called STX17, and we've talked about this. Heterozygous and homozygous individuals for this mutation have a gray coat color, and that penetrance is 100%. So over time with aging, all of these horses will become gray. The other thing that we've known for a very long time about gray horses is that they have an increased risk for melanoma. And while melanoma in and of itself is very rare, in the general horse population, greater than 75% of gray horses over 15 years of age develop melanoma. So once the gray mutation was discovered, a really nice work was done to show that gray is also a risk allele for melanoma, and that if you have one copy of the gray mutation, you have a 1.06 increase in melanoma grade. So not only are you more likely to get melanoma, but the severity of your melanoma is higher. If you have two copies, that increase in severity is 1.22 melanoma grade. So suggesting that melanoma penetrance is incomplete in gray horses because not all of them develop melanoma, and additionally that the gray gene is actually a risk allele for the development of melanoma. It doesn't stop there as far as complexity for melanoma risk in gray horses, unfortunately. So we know that the STX17 gray allele is a risk for melanoma, and it's a gene of major effect. But we also know that melanoma grade is modified by mutations in the ASIP gene, which happens to be the coat color mutation that controls whether horses show a bay or black phenotype. And that horses that are heterozygous or homozygous also have an increased melanoma risk. And so when we start adding these things together, in my mind, while gray is a simple trait, risk for melanoma is actually a more complex genetic disease. And so it makes sense to just stop for a moment and really define complex or multifactorial genetic disease. And so just as it sounds, this means that disease expression is complex, and that inheritance of disease or disease risk may be polygenic. So with gray, your inheritance of melanoma risk not only is dependent on your gray locus genotype, but also your ASIP locus genotype. And I'm guessing within a couple of years, based on further research that's going on, we're going to know that that melanoma risk is also dependent on genotypes at other loci. Diseases are also complex because they have environmental influence. And the third thing that makes them complex is that the phenotype may be quantitative. And so instead of an answer of yes or no, you either have this phenotype or you don't. Phenotypes for complex traits are often measured in things, often measured numerically. And so an example of this in people would be, for example, body mass index, or insulin levels associated with diabetes. Those would be a couple of examples of quantitative traits. And so, as you can see, we're moving into a realm of things that are much less clear-cut as far as being able to interpret them. So I always think of these things mathematically. For me, the equation is phenotype, or the resulting phenotype, is the sum of the effect of your genes and your environment, plus something else that I term individual variation, and these are things that we probably can't measure that have many effects. That genetic background idea and individual variation would be important. So really complex traits are a summation of all these things. So again, we've alluded to what are the complex genetic diseases in horses, but I just put them here to reiterate those things. Some of the ones that are complex and currently being looked at in the horse are Equine Metabolic Syndrome, in particular with reference to susceptibility of developing laminitis, and we'll talk a little bit more about that in a moment. Recurrent exertional rhabdomyolysis, and so I saw that someone had a question about this disease, and at the end we'll come back and discuss RER and PSSM and compare and contrast. Osteochondrosis and osteochondrosis dissecans, these are developmental orthopedic diseases and cartilage abnormalities that we often see in young horses, particular joints, fetlocks, hocks, but also wobbler syndrome is probably a manifestation of OC and OCD. Recurrent airway obstruction, or HEAVS, we know likely has a moderate to major genetic component. And then some immune-mediated diseases. One that we're looking at in our group is equine recurrent uveitis. There's a lot of really good examples in human medicine of immune-mediated diseases with genetic components such as rheumatoid arthritis, Crohn's disease, and others. So when we start talking about genetic testing in complex diseases, we're no longer talking about yes or no answers. And when we're talking about genetic research in complex diseases, again, this isn't yes or no answers. So our goal is not to necessarily identify a simple genetic test where we can test horses and decide to breed or not breed them. Our goal really here is to identify the alleles that result in genetic risk. When we do that, we can do two things. We can quantify the risk for each allele, which means that we can do a genetic test in individuals and quantify the risk for that individual developing disease. So what does that mean? For example, if we had identified genetic risk alleles for equine recurrent uveitis, and we know that there's at least 2 of them, we can take an individual, determine their genotype at those 2 risk alleles, and try to assign some kind of number to their risk for developing disease over their lifetime. So we might say that this individual horse has a 25% chance of developing equine recurrent uveitis over its life, whereas this individual has a 50% chance, or maybe this individual has a 75% chance. Well, why do that? Why is it important to quantify the chance that they're going to get diseases? Well, as you can imagine, there's lots of reasons to do that. One of the simplest things might be making a decision about, do I want to purchase this horse? Or do I want to breed this horse knowing that their offspring have a high risk for disease? As a veterinarian, in my mind, one of the things that I think is really important for quantifying risk of disease is for preventative medicine. And so I want to be able to look at horses when they're born and say an Appaloosa horse, which we know that breed has a lot of equine recurrent uveitis, test that individual horse, look at its genotype, and be able to look at the owner and say, this particular foal has a very high risk for developing uveitis during its lifetime. And because of that risk, we're going to do these things. With the example of uveitis, it might be, we're going to make sure that a veterinary ophthalmologist examines this horse at least once a year. And at the first sign of disease in this horse, we do intervention. So that might be putting the horse on immunosuppressive drugs, doing some kind of surgical intervention in the eye where we actually put implants with immunosuppressive drugs in, something like that, with the goal that this horse will never actually develop disease and will never suffer from its disease. With EMS, the example might be with metabolic syndrome. We know that this horse is at high risk of having metabolic syndrome, so we're not going to let him or her graze early spring grass pastures when we know environmental effects are there. Similarly, for PSSM, we can say this horse should never be fed a high-starch diet. The classic example in human medicine would be the BRCA1 and BRCA2 mutations that identify individuals at risk for breast cancer. And this is used routinely, that when individuals are tested and have one of these mutations, they have early and more frequent screenings for breast cancer, particularly in women, but also in men, because it does increase the risk in them as well. So we've talked a little bit about metabolic syndrome. This is a disease that's a hot topic. It's a disease that we're working on in our lab, and it is probably, in my mind, the classic equine complex disease, and it has a lot of similarities to the classic human complex diseases of type 2 diabetes and also obesity and insulin resistance. So what is equine metabolic syndrome? Most of you are probably familiar, but just by way of brief review. These horses tend to be obese. That can be generalized obesity, that they have very high body condition scores, but also regional obesity. And the classic thing is the crusty neck. So you can see this horse here and this little pony have a crusty neck. They also have hyperinsulinemia, or high insulin levels, and insulin resistance, and an increased risk for laminitis. And so there's some debate in the equine metabolic syndrome community about whether laminitis is a criteria for calling a horse EMS or is it a result of EMS. But definitely this kind of combination of clinical signs and clinical syndrome goes together. We know that from a lot of research that's been done, particularly in the last several years, that diet plays a huge role in obesity and insulin resistance in people and also in horses, and there's a lot of great data that our collaborators have put out there that show diet is important. But what we also know is even in the same eating the same diet or having the same exercise regimen, which is clearly plays a role too, some horses will have metabolic syndrome symptoms and some will not. And so what we think the difference is here is really genetics. And so I've kind of shown this diagram to show that those horses that have disease are overlapping typically in a couple of these categories, if not all three, and they all not only tell us something about an individual's risk, but they also tell us about disease severity. So what are the goals of EMS genetic research? And so this is really reiterating what we said before. In our research with EMS, our goals are to identify one or more underlying genetic risk factors, number one. So we can start along this process of identifying individuals at risk. And then number two is to understand how those specific genetic risk factors actually interact with environmental risk factors. So we know, for example, that grazing on spring pasture can result in metabolic syndrome-like signs, particularly resulting in laminitis in these horses. But is it just the amount of energy in pasture itself? Are there specific components in pasture? Are there things that we can do to better define what's going on and what the interaction is? So from there, we can develop a genetic test to predict an individual's disease risk, but we can also develop a more intelligent and specific disease prevention plan for at-risk individuals. So it might be eliminating one specific dietary component. It might be actually choosing one drug intervention, one pharmacologic intervention that makes sense based on disease pathways, or it might be doing all of the above. And so really understanding the genetics and understanding the disease and understanding these interactions, in the end, hopefully will lead to better care for horses and healthier horses. So other complex traits, I've talked a lot about disease, and as my training as a veterinarian, genetics and disease are on the forefront of my mind. But we're also seeing with the increase in genetic tools that people are more and more looking at other phenotypes of interest, and most notably phenotypes that are associated with performance. And so I'm guessing that many of you may be familiar with the thoroughbred speed gene test that's become commercially available in the last year. So this is based on a single base pair change in the non-coding sequence of a gene called the myostatin gene. And myostatin contributes to many muscle phenotypes. This locus, or this gene, was identified as being important in racing performance in thoroughbred horses by 2 independent studies. So the first study was of 118 elite thoroughbred racehorses that were from Great Britain, Ireland, and New Zealand, basically. The second study was in 189 elite thoroughbred racehorses in North America. So the important thing to recognize in both of these studies is that this gene was shown to be associated with optimal racing distance. And so it's considered the speed gene, and it's the speed gene test because having this mutation in myostatin is associated with short distance, and there's some small amount of data in one of the papers that say that some of these short distance horses may have better earnings, although it hasn't been really sorted out whether that's because these horses, who they were trained by, or that they were trained for shorter distances, or these types of things. And the other thing that's important to note about these 2 studies, and some other subsequent studies have come out, is that they don't agree on what the underlying functional allele is. And so the speed gene test is for a non-coding sequence change in myostatin. That SNP itself has been associated with racing distance, but it potentially may not be the underlying biologically functioning allele. And so there still needs to be more work done on this. So what does that mean for interpreting the test? Well, it means that it's somewhat hard to make a direct correlation between genotype on the speed test and what a horse's ultimate race performance will be. We might be able to say something about what distance the horse is likely most suited to run, but whether or not he's going to be a top performer or a top winning horse is not necessarily a clear conclusion that can be drawn from this test. And that really goes for all performance tests, and similar with disease tests, is that performance is a highly complex trait. And so when you're thinking about testing for performance, you want to make sure what the definition of that performance is. So the speed gene tests for racing distance, and the definition of performance in that test is really racing distance, and not necessarily how fast the horse is going to cover that distance. And the data on that, how fast piece, is probably forthcoming. Also, that optimal racing distance for a horse is going to be affected by many things, not only muscle phenotype, but also cardiovascular phenotypes, particularly respiratory phenotypes, and other things. And that the environment and the training that an individual horse has is going to play a huge role in what the expression of that phenotype is. And so just to reiterate that it's complex, not saying that information can't be gained from this test, because certainly there's clear published papers showing that it does tell you some information about a phenotype in horses. It's just how to interpret that phenotype, I think, will become more clear as more information is gathered. And that being said, think I've mostly said this, but single gene tests are one piece of the information, and this is really an ongoing area of active research, and I'm guessing we'll have quite a bit more information over time. So I've talked a lot about complex genetic traits. Hopefully I've kind of gotten the point home that interpreting them will be difficult, and they're certainly not going to be yes-no answers, and that decisions based on single gene or simple genetic tests need to be taken in light of all the information about disease or performance traits. I want to switch gears a little bit and talk about the advances in equine population genetics, because this is another area in the last little bit I want to focus on, because I see the genetics community heading in this direction. So population genetics is really the study of the genetic composition or the frequency of allele in populations. So thus far we've been focusing on the alleles that occur in an individual. Now we're going to shift gears and say the alleles that occur in populations and the frequencies of these alleles that occur in populations. And in horses, when we're talking about populations, 99% of the time we're talking about breeds. And so what are some of the population genetic questions that the community is trying to answer? The two biggest ones, and I'll share a little bit of data with you about these first two questions, are what is the genetic diversity within a breed? And so these are questions like, what are the level of inbreeding? How much variability is there between two thoroughbreds? How much of their genome do they share? How much of their genome is different? And the second is what are the genetic relationships between the breeds? We certainly, for many of our horse breeds, have historical records about what breeds they were formed from, what breeds have influenced them. But now we actually have the tools that can help pick out, based on genetics, what really was happening 200 years ago, 500 years ago, 2,000 years ago as far as breed genetics. And sometimes we're learning that the breed histories as written are not necessarily correlating with the genetics that we can see in the horses that are living today. So one big push in the equine genome research community that's actually being headed up by our group here in Minnesota is the Equine Genetic Diversity Consortium. And so this is a group of greater than 30 scientists worldwide from 22 different institutions actually, who've contributed genetic data from 35 different breeds. And in this set of over 35 different breeds now, we have now greater than 1,500 horses. And every one of these horses has been genotyped for 54,602 genetic markers. And these genetic markers we call single nucleotide polymorphisms. And so we call them markers because the majority of them do not occur in a protein coding gene. So remember earlier we limited our discussion to protein coding genes. Here we're just looking at genetic variation in the genome. These markers are linked to protein coding genes, so we can make some conclusions about phenotypes from this data, but this is not specific genetic mutations that we're testing for. So what can we do with this data? Well, we've gathered all these data from these horses, and our group have done a couple of things. One thing, and this is a paper actually that should be coming out later this year, is we've looked at how inbred are different horse breeds. And so we use this data to kind of calculate a relative inbreeding coefficient from breeds. And so what we have here listed in this table are the horse breed and this computation of inbreeding coefficient. So this is the actual number, but we can actually convert this into percentages and see, for example, in Mongolian, their inbreeding level in the genome from these horses was about 1.5%. In contrast, the inbreeding level in the thoroughbred from the sample that we looked at was about 15%. And these breeds are actually organized by level of inbreeding. So Mongolian horses, which are a kind of large, extensive population of horses in China, and the quarter horse are two of our least inbred horse breeds, whereas the thoroughbred and the standardbred are two of the most highly inbred breeds based on the breeds included in this analysis. So other interesting things to pick out from this, well, the quarter horse, some people might guess first off based on breed history, might be highly inbred because they're a relatively young breed and they were started from a relatively small set of founders. But one of the interesting things about the quarter horse is that those founders of the breeds were highly admixed. They had a lot of different genetic origins, which led to a lot of a lot of genetic diversity at the input of the breed. And the other thing to remember, the quarter horse, and this goes for the Hanoverian, some of the other warm blood breeds as well, is that they allow admixture into their breeds. So what's admixture? Admixture is a genetic term which basically means crossbreeding. So the addition of thoroughbred bloodlines into the quarter horse and allowing those horses to be registered would be an example of admixture. In human genetics, the classic example of an admixed population is African American populations, which have both Northern European and African origins. And there tend to be more genetic diversity in admixed populations. The Mongolian horse probably has a lot of genetic diversity for a couple of reasons. One, it's a very old horse population, and so there's been a lot of time for diversity. And two, it's a very large horse population. So there's about 4 million Mongolian horses. There's about 3 million quarter horses. And so you can imagine in that large population and breeding in a large number of horses, there's just more room to have different combinations and more diversity. The second question that we've been trying to answer, of many, but some that I can show you some data from, is what's the relationship between horse breeds? And so what we have here is another graph. And in this graph, what we're looking at is something called principal components. So we took the genetic data from each horse across all 54,000 markers, and we mathematically tried to simplify that data with something called a principal components analysis. And so we have plotted on the y-axis our first principal component, and on the x-axis our second principal component, and the values. So it's not important really here to understand the complex mathematics, but to understand that each principal component is a way to capture the variation in genetics between individual horses, and that the first principal component captures the largest amount of variation, and the second principal component captures the second largest amount of variation, and so forth. And we've actually computed up to 20 principal components in this data set, but here I'm only showing you 1 and 2. And so we find some interesting things from this data. Again, this will be in a paper that should be published later this year. But we see, for example, that breeds, for the most part, tend to cluster together. And so each one of these pink squares is an Arabian horse, and all the pink squares are the individuals of the Arabian breed that we tested. And for the most part, the Arabians cluster together. We have this one Arabian horse that's standing out here a little bit, and that's because this was a horse that was used as part of a sequencing and SNP discovery effort. I won't go into that, but this would be expected. But breeds tend to clump together. So here's the Arabians. Here in black we have the Andalusians. Over here we have 3 different colors. These are actually 3 different sources of thoroughbreds, which is kind of interesting. Some from the United Kingdom, some from here in North America, the University of Minnesota, and actually some from Ireland. But despite the fact that these thoroughbreds were from 3 different geographic locations, the thoroughbred is a pretty pretty homogeneous breed. So again, the IRE are thoroughbreds, are thoroughbreds, and UK are thoroughbreds. So there's kind of a brownish color, a blue color, and a turquoise color down in this kind of lower right-hand corner. I'm not sure if you can see my mouse, I'm circling them, but lower right-hand corner of the graph. The other interesting thing that we found from this graph is that there are some breeds that don't cluster as tightly together. And so in red, an example of this would be the quarter horse. And so they're a little bit spread out across the x-axis. You see a few individuals are spread out here. Another example would be kind of these burgundy boxes in the same area with the quarter horse that are actually the Hanoverians. And that's because these are admixed breeds. And so what's interesting is both the quarter horse and the Hanoverian kind of spread out over towards the thoroughbred breeds, and that's probably due to the thoroughbred influence in these breeds. And when we look at these data in many other ways and many other graphical displays, we see those things. We can actually pick out thoroughbred, for example, thoroughbred influence on some other breeds. Another thing to point out is historically we know that there was Arabian influence in the thoroughbred. It's written about, it's talked about how strong that is, but there's been enough separation between these 2 breeds over time that the Arabians up here in pink are actually separating by distance, genetic distance, quite far away from the thoroughbreds here with their multiple colors down here in the right-hand corner. So just a few interesting things to pick out. We could spend an entire hour talking about this kind of data and all the different displays, and many conclusions and further investigations are going on in this data, but it's kind of fun to think about what can be done with this type of data. So for the sake of time, I'm not going to talk a lot about more population genetic data, but I just want to throw out there kind of the two questions that we're looking at that I think are quite important, and I would guess we'll have quite a bit of data published in the next couple of years. One is, what are the impacts of artificial selection or human-directed breeding in horse breeds? And so one of those things we see is decreased diversity. Certainly inbreeding, lowering diversity in a breed sometimes can be good because it helps us fix phenotypic traits of interest that we want to represent the breed, but on the other hand can sometimes be bad because it can also fix deleterious alleles or increase the frequency of these. Sometimes with human selective breeding, we even inadvertently select for deleterious alleles. We may be selecting for a gene that confers a phenotype of interest, and there's a mutation sitting close to that gene, and by genetic hitchhiking we pick for it. Or we may believe that a disease mutation can cause a secondary good phenotype. That's a little bit of the example with Overo and with leopard complex. The other big question that is a effort of the Equine Genetics Diversity Consortium is to really understand what are the alleles that are driving phenotypic differences between breeds that we're actually selecting for. For example, what are the alleles that make a Tennessee walking horse and make a Tennessee walking horse express their very unique gait? And what are the alleles that make a draft horse and large in size, or a miniature horse and small in size? So those are the kind of questions that we're heading to. So I know that you guys have been typing some questions on the chat, and I want to make sure we have time to get to them. So I'm just going to wrap up really here quickly with conclusions to say a couple of big things. Genetic testing for simple traits is increasingly available. The future of equine genetics is really moving towards more complex traits, and so while I'd like to say it's going to make everything simple, the more information we have, it's probably going to muddy the waters, and it's going to take people that understand genetics and are skilled in interpreting tests to really help with recommendations. And three, that these new genetic tools are really allowing us to do more in horses and more sophisticated investigations, both on the disease front but also on the population genetic fronts that we've never done before. So what we know about horse breeds and horse genetics really is going to increase dramatically. So acknowledgements, a lot of this data from many collaborators, I won't read them all off for the sake of time. You'll have these slides. But then also just to acknowledge Equine Genetic Diversity Consortium, which there's a lot of people, a lot of different places, and you can find more information about this on our website too. So with that, I'm going to kind of go through the questions that everyone had. So the first question is about the champagne gene, and I'm going to leave that one aside for a little bit just simply because coat color testing is not my #1 area of expertise. I know this test is commercially available at the University of California at Davis. And I know that the publication has recently come out, but I'm going to set it aside for a moment because I think Davis has got quite a good information on their website, and I want to get to some of the stuff that I maybe can shed more light on. The first one is, excuse me, differences between PSSM and RER, and the second half of that question is RER more likely than PSSM in Arabians? So the first half of the question is PSSM PSSM and RER are similar in that these horses have similar clinical signs, and that tying up is the most common clinical sign. PSSM is a glycogen storage disease, and so there's more than one form of PSSM, and I only talked about type 1, but both type 1 and type 2 PSSM result in excess glycogen storage in the skeletal muscle. RER, or recurrent exertional rhabdomyolysis, in contrast, we think is a disease of abnormal muscle contractility. And so that disease is first described in the thoroughbred. There's been a recent paper describing it's very similar in the standardbred. We also see it in quarter horses and Arabians. So both genetic diseases both result in tying up, but very different underlying disease biochemistry and pathophysiology and different underlying genetic causes. With RAR, we're working in our lab actually on trying to understand the genetic basis of this disease in the thoroughbred and in the standardbred. We've made progress, but no genetic mutation to date. And again, as I alluded to earlier, it's probably going to be complex. I'm guessing we'll have more than one risk allele for RAR. The second half of the question is RAR more likely than PSSM in Arabians? Type 1 PSSM we have not to date identified in Arabians. We have, based on muscle biopsy, identified type 2. But RAR is also in Arabians. Relative frequency of the two diseases, I would say that RAR is more likely, but both of them would be best diagnosed by muscle biopsy. And so really the same diagnostic test for both those diseases if you have a horse where there's a question. The next question is EMS found in wild horses? And do our environmental practices and breeding contribute to the high incidence of the disease? So I have spent a lot of time thinking about EMS and the environment. What I believe is happening with EMS, which I didn't have time to talk about today, is that the underlying genetic susceptibilities to EMS, I believe, are probably occur in wild horses at some underlying rate. And historically, one for wild horses and historically for domesticated horses, before we started feeding them lots of high starch feed and putting them in stalls and really giving them dietary excess, may actually be advantageous, because the alleles that underlie metabolic syndrome are probably also alleles that make horses very metabolically efficient and very, very good at utilizing feed and very feed efficient. And so I think that with metabolic syndrome, that we will find, when we identify genetic alleles, we will find that they segregate in wild horse populations, which are actually feral horse populations, and that a huge impact of environment on the risk for developing disease. I think that we may even find some breeds of horses, such as the Morgan horse and maybe some of the pony breeds, where almost all horses have the genetic risk, and a huge part of disease expression is environment. So hopefully that answers that question. The other question is the role of environment in the development of cerebellar abiotrophy and other issues in Arabians. I mean, I think that I think we don't understand enough about the genetics and environments. I think they're probably more likely to be simple diseases, but I don't know that we won't necessarily find other genetic traits or environmental contributors. I think that the data is still in its infancy in these traits. They're probably tending to be more simple, but I think it's hard at this point in time for me to draw a conclusion on that. The final question is another webcast dedicated to population genetic studies. I think it's certainly a possibility. Those studies right now are in a relatively early stage of collaboration with a big group of people, and actually there's a Havemeyer meeting of the equine genetics community next week, and one of the things that we'll be discussing is where we want to go. But I would think that we'll have quite a bit of data about genetic diversity that will be published within the next year or so, and so certainly would be open to doing a webinar, kind of sharing some of that data, because it is actually really tremendously interesting. We're finding a lot of really interesting things. So with that, I think I'll try to wrap things up. And if anybody has other questions, there's a lot of information about a lot of these diseases on our Equine Genetics and Genomics Lab website. So thank you very much.
Well, I just want to take a quick second to say thank you so much for that presentation, Dr. McCue, and thanks to all of you for participating and asking great questions. Later this week, you'll probably receive an invitation by email to participate in an online survey about today's webcast, so if you could take a few minutes to give us your feedback, we would appreciate that. And the next webcast in this series on equine genetics will be on October 18th on the topic of EMS in ponies, and if you check our website, myhorseuniversity.com. You'll find details there and the registration link. And our My Horse University and Horse Quest free monthly webcasts will return in September, and details are on our website for that as well. And you can become our fan on Facebook or follow us on Twitter for more up-to-date information about events and promotions. And also a reminder that this webcast was recorded and will be uploaded to our website by the end of the week. And you can send us your comments and suggestions to info@myhorseuniversity.com. So thanks again and have a great afternoon.